Thank you guys for inviting me. Thanks very much to David and Mary and Helen for making this happen, and Yisong and Lior. My impression is this bootcamp is pretty machine learning and data analysis oriented. So for my first introductory lecture on the first day, I went with something pretty basic sciencey just to give a sense of where some of these analyses are coming from. Then we'll get into the actual model fitting and analysis later on. So I'll start with a vocab word. So a helpful word for when you're attending seminars. This is, why am I not talking? So when you're attending boot camps like this, it can be a lot of things happening and you can feel kind of overwhelmed. Everybody's coming from a different background. So if you feel like you're not following what's going on, or just not seeing the point or anything, ask yourself why you're not talking, and please feel free to speak up. I mean, this is a boot camp and a discussion, so you should feel free to ask questions and raise concerns about the analyses that we're presenting. I mean, it's a small group, and the whole point is to get comfortable with these models and understand not just like the material that's on the slides, but how it's applying to your research and how you can kind of internalize that knowledge and make use of it. So please do feel free to speak up over the course of the talks today and during the hands-on session. And I hope that you're able to get something useful out of this, even if some of the stuff I show isn't necessarily what you work on in the lab. I hope that it has some benefit to you for your understanding. So the theme for today is dynamical time series. And the reason that I wanted to start out with time series analysis is because basically everything in neuroscience is about time series. You always have an animal that's running around and interacting with the world, and you always have to make sense of what the brain is doing over time in terms of what the animal is doing over time. And this is something that makes it a little bit different from some areas of machine learning where you're working with image recognition or general like signal processing data. Neuroscience, there's always going to be a temporal aspect to cells' responses, and we want to understand what the biophysical properties of neurons do to shape their responses and to shape this transformation from stimulus inputs to recorded neural activity. And some of the things that individual neurons and neural populations can do as an animal's running around and interacting with things in the world. So this is true at the level of individual neurons. Neurons receive input from stimulus features in their environment, filtered through other neurons in the brain, and produce spikes occurring over time in response to that stimulus. It's also true at the level of neural populations. If you have a cortical column or a pool of neurons in a nucleus of the brain, and they're all interconnected with each other and they're firing, and you feed that neural network an input, those inputs are going to evoke a diverse range of responses in recorded neurons, depending on how much input they're getting from this external source versus how much input they're getting from other cells in the neural network. And you often see these diverse, temporally complex, filtered versions of the input in the responses of individual cells. And one of the advantages of this temporal complexity is that you can have readout neurons elsewhere in the brain that can sum together the activity of different individual cells and produce arbitrary transformations of their input. So there's a nonlinearity here, not just in scaling the input to the neural population, but also shifting and transforming that signal and stretching it out over time. And this is something that's really fundamental to individual neurons and neural populations, is that they apply this temporal filtering to their inputs that they receive over time, and that is a big part of what allows our brains to do the things that they do. They have to integrate information over time, they have to transform signals, and they have to shift signals in time in order to carry out working memory tasks and produce time-evolving motor outputs and any number of temporally structured things. So when you're dealing with time series data in neuroscience, there's kind of two families of tools that you'll encounter. Broadly speaking, dynamical systems tools, which are more maybe for modeling the responses of neurons to inputs and understanding how they transform their inputs over time, and also statistical and signal processing models for making sense of what a neuron's responses are doing to the input to that cell, and just how you summarize that transformation from input signals to a neuron to their firing rate outputs. In the area of dynamical systems, you can use models that describe the behavior of neurons based on first principles, interactions of ion channels in their membranes, processing of inputs over time. And you can think about what neurons are doing to their inputs at different levels of abstraction for different purposes. And you can take these dynamical systems models and you can analyze them to find interesting structure and interesting dynamics at the level of single cells, at the level of neural populations, that kind of tell you how these neurons or pools of neurons are operating on their inputs. On the statistical side, more practically, oftentimes we want to just summarize what our recorded cells are doing. You stick an electrode somewhere in the brain and your cell is spiking, the animal's doing something. And you want to just come up with a quick summary of what the relationship is between those two things. What features of the stimulus is that neuron responding to, and how is it transforming those features, and how is it filtering them? So these are kind of the two flavors of time series analysis and modeling in neuroscience. And this morning, we're going to focus mostly on the first of these, dynamical systems models of neurons. And this afternoon, in the practical session and the hands-on component, we'll be looking at one tool for making sense of recorded neural activity and describing neural responses in terms of their inputs. So that's the goal for today, starting with some more abstract dynamical systems models and going to something that you will actually probably be able to use on your data sets. Any questions so far, or comments or anything? Okay. So we'll start with the dynamical systems portion of the class, and to do that, we're going to start with an equation. Can anybody tell me what this is? Yes, okay. This is Ohm's law. This is the relationship between voltage, current, and resistance across a capacitor. And this guy? Yeah. Less common. This is just an equation for the dynamics of a capacitor, how the current across a capacitor is a function of its capacitance and the rate of change of voltage across the capacitor. So if you know these two equations from your freshman undergrad electricity and magnetism course, then you can do a lot of modeling and analysis in neuroscience, and you can get yourself pretty far just based on these two concepts and thinking about how to apply them to what neurons in the brain are doing. So I'd like to go through building up these concepts into a model of what a single cell is doing when it receives injections of current and what the different currents across the cell membrane are doing. And then we'll talk about ways of simplifying that down and interpreting these dynamical models. So a core property of neurons and also of all cells is that they store energy in the form of a charge differential across their membrane. So the membrane of all cells is this lipid bilayer of charged outer heads and these hydrophobic tails in the middle. And any ion or charged particle is going to have a really difficult time crossing this lipid bilayer. As a result, you have this effective separation of the internal contents of the cell from its external environment. And this allows you to build up a charge and a concentration difference across this membrane. So if you think about this and you think about electricity and magnetism, you might realize that this is pretty much acting like a capacitor. So a capacitor is a set of charged plates with an insulating material in between those plates. And when you deposit charge on one of the two plates, it's unable to cross to the other side, so it induces a charge on the opposite side of the plate. Similar to in a lipid bilayer, you can have charged particles outside and inside the cell, but it's very difficult in the absence of tunnels to pass through this bilayer and distribute charge across the membrane. So this is a way of storing energy because you can push ions to one side of the lipid bilayer or the other, and the natural tendency of those ions is to want to flow down their concentration gradient or their electrostatic gradient to where the concentration is lower or where they're attracted as a charge point. So the way that ions are able to flow across the membrane of cells is by passing through ion channels. So these are protein structures. This is probably very familiar, but these are protein structures. They allow only a specific ion through them based on the shape of the channel. They have binding sites that can only accept an ion of the right radius and the right charge. And they allow the flow of ions from one side of the cell membrane to the other. So in keeping with our work of relating these biological properties of cells to properties of electrical circuits, you can think of this sort of as a resistor. It allows charge to flow through it, but there's some inherent resistance. There's only so many ion channels that are available. Ions can only pass through them at a certain rate. So the more ion channels you have in your membrane, the lower your resistance is, the easier it is for a charged particle to flow from one side of the membrane to the other. The fewer channels there are, the higher the resistance of the membrane to that particular charged particle. So there are two forces acting on charged ions in neurons. There's a concentration difference and a charge difference. So if you take this cartoon diagram here, the positively charged ions, get a laser pointer. The positively charged ions want to flow across the membrane in this direction for two reasons. First, it's a lower concentration inside the cell versus outside. And second, they're positively charged, so they're attracted to this negatively charged population of ions inside the cell. And for any different ion that you have in the neuron and in its surrounding environment, you have these combined forces, the concentration gradient and the electrostatic gradient, that are acting on the ions. And if you take these two forces and apply them to the ions and just allow them to equilibrate, you'll come to what's called the Nernst potential, which is kind of an equilibrium point where the force acting on the ion due to a concentration gradient and the force acting on the ion due to a charge difference are balanced out and equal. So for each individual ion, you'll have a different Nernst potential, which is the balance of concentration of that ion inside and outside of the neuron. So what this Nernst potential does is it is effectively giving us a power source to our model. It's giving us the driving force with which ions are transported across the cell membrane, and that force is proportional to the membrane potential, the difference in voltage across the cell membrane, and to the concentration of ions inside and outside of the cell. So if we take these elements of the cell membrane, we have three components. We have a capacitor, we have a resistor, and we have a power source. And if you take these three elements and you hook them up together, what you get is a very simplified model of what a neuron is doing. You have the region outside the cell, the region inside the cell, and you have flow of charges through this circuit over the course of time. So this is a simplified diagram of what the membrane of a neuron is doing. We can now take this diagram and make it into a mathematical description of neurons by bringing in the two equations that we looked at earlier, Ohm's law and the capacitor dynamics equation. So we have a rule that relates the voltage drop across a resistor to the current passing through it and its resistance, and we have a rule for relating the capacitance of a capacitor to the current flow across it and the change in voltage over time. So with these two equations, we can describe what's going to happen to charge differences across this neuron and how its membrane potential is going to evolve over time. So to do this, what we're going to do is make a couple substitutions and simplifications. We'll take Ohm's law V equals IR and convert it to be something in terms of current, just by dividing both sides by the resistance. And we're now going to make a pair of simplifications. We're going to replace resistance with conductance, which is just 1 divided by the resistance. So instead of saying how much resistance there is to an ion flowing through a channel, you're saying how readily that channel conducts ions into the cell. And we're going to introduce this Nernst potential of each individual ion. So this is the force that is acting on the ion and trying to push the voltage drop across the cell membrane to a particular voltage, say a -80 voltage or a -60 voltage or a -50 voltage. Each ion kind of is happiest at a different membrane potential difference across the cell. So if we take this equation for the current induced by a particular ion and its differential concentration across the cell membrane, and we plug this into the capacitor dynamics, we get something that looks like this. So this is just plugging in for I, this term that we derived over here. And now we have the capacitance, and the change in membrane potential across the cell is a function of the conductance of the cell and the difference between the cell's membrane potential and the Nernst potential of our particular ion. And with a little bit of rearranging, we can come to a formulation that looks like this. We've just divided both sides by the conductance and turned it back into a resistance, and we get this equation that looks like this. So we have our equation that is describing the behavior of this little simple passive membrane of our cell. And we want to understand how it's going to behave. The first thing that we can do is we can say, what is this going to do if we let time run out infinitely? So this is an equation for how the membrane potential is evolving over time. This is just the update rule, dV/dt, of the membrane potential V. And as you simulate forward in time, you're eventually going to converge to a particular value of V as you update its value according to this dynamical equation. And we can find that just by plugging in 0. We're finding the point where the rate of change of the membrane potential is 0, and solving this equation for V. And that tells us that the steady state membrane potential of this simple model that we've made is just equal to the Nernst potential of our particular ion. So this is telling us that if we take this model and we start it at any arbitrary membrane potential, it's going to evolve over time, and it's eventually going to converge on this reversal potential of our model ion. So this is what you would call the steady state value of the steady state value of the membrane potential. And we can also, from this differential equation, understand how the membrane potential of the neuron is going to approach this steady state value. So we know that it approaches it at a rate that's proportional to this product of the resistance and the capacitance, because this R times C term is just scaling the rate of change of the membrane potential here. So the resistance and capacitance of our neural model is effectively setting the time course with which the membrane potential of the neuron increases or decreases to its steady state Nernst equilibrium potential of our model ion. We also can tell from this differential equation that it's going to approach it exponentially. This is just something that we know from differential equations, that if you have an equation of the form dx dt equals some function equals x, then a solution to this differential equation is that x of t evolves exponentially and decays exponentially to its steady state solution. You can derive this, but I decided to just skip over it here and focus on kind of this just magical fact that if you start out at any arbitrary condition, you will exponentially decay down towards your Nernst potential. So what this looks like in practice is if you take a neuron that's getting no external current and you inject a fixed current into the neuron for some period of time, here's the applied current here, the membrane potential is going to not instantaneously jump up, but it's going to slowly approach the steady state value of this dynamical system. So the steady state value is the reversal potential plus the current scaled by the resistance, which you can find just by dividing this term by R and solving for dV/dt is 0. And the rate at which it approaches this steady state value is again equal to the resistance times the capacitance. So this gives us what's called the membrane time constant of the cell, the rate at which the dynamics are evolving over time. And in this case, there's just a single ion and a single resistance that's affecting this. So you get this nice simple equation for the time constant in terms of these two things. So in general, in neuroscience, most of your neurons are going to behave like this. They're going to receive a step input, and they're going to show some time evolving response to that input as they approach a new value of their membrane potential. So the other thing that makes neurons interesting and makes them not just passively responding to these injected currents is that they spike, they generate action potentials. And the way that they do this is that on top of having passive ion channels with a fixed resistance, they have voltage-gated ion channels where the resistance of the ion channel to the flow of ions is proportional to the membrane potential here. So in practice, how this works is you have a charged particle that's attached to this ion channel, and the membrane potential across this lipid bilayer is going to determine how this charged particle is pushed, whether it's pushed further into the cell or closer into the lipid bilayer. And as that charged particle is pushed, it changes the configuration of the ion channel to go from a closed configuration where ions can't flow and resistance is very high to an open configuration where ions are allowed to flow through freely. So in our model, you can do this just by replacing this simple resistor with a variable resistor, where now the resistance of your ion channel is a function of your membrane potential. So we've introduced membrane capacitance, we've introduced Nernst potentials, we've introduced resistances of membranes to flow of ion, and we've introduced voltage-gated ion channels that have a variable resistance based on the membrane potential of the cell. Are there questions at this point before we go further? This might be something you guys have all seen before, but I wanted to do something that was a little more grounded in the biology and talk about how it's applied in theory and modeling. So is this something that folks have seen? Yeah? Okay. So this famously was the basis of the Hodgkin-Huxley model, which was published in Journal of Physiology in 1952. This is the summary diagram of the electrical circuit representation of the cell membrane. Hodgkin and Huxley added in 3 of these channels, one for sodium, one for potassium, and one which was just a leak channel, which is kind of a catch-all term for any other electrical difference across the membrane of a cell. 2 of these ion channels, the sodium and the potassium, are voltage-gated ion channels. So how open or closed the channel is a function of the membrane potential of the cell, of the voltage difference across the cell membrane. So very quickly going through membrane potential dynamics in the Hodgkin-Huxley model. All that we're doing now is we're adding in additional ion channels for these different ions in our system. We have sodium, we have potassium, and we have leak. So starting at our equations for our simple passive neuron model, we're going to expand out the current terms of our capacitor dynamics to include the current due to sodium ions, the current due to potassium ions, and the current due to the leak ions, which is just anything else that's present. And we'll also still allow an external current to be injected by an experimenter or by another neuron. And expanding out these individual terms, what we end up getting is something that looks like this. So it's really just, it's a lot less scary than it looks. It's 3 repeats of this conductance term times a difference between, I might have a sign flip there, a difference between the membrane potential and the reversal potential of each individual ion. So there's a term for sodium, there's a term for potassium, and there's a term for the leak. And importantly, both the sodium channel and the potassium channel are voltage dependent. So the conductance of the channel is a function of the membrane potential of the cell. So this might be very high when you're at a very low membrane potential, and as you increase the membrane potential, the channels are going to open, your conductance is going to increase, and now these 2 terms are going to contribute much more strongly to the dynamics of the cell. So the extent to which these 2 terms contribute to the dynamics sets the fixed point of this model, because if these 2 conductance terms are 0, you get pushed to the leak reversal potential, and if these 2 terms are non-zero, then you're getting pushed to some combination of the leak, the sodium, and the potassium reversal potential. So what this modeling of the dynamics of ion channels gives us is an ability to capture the actual spiking dynamics of neurons when they're generating action potentials. So if you take your model neuron and you give it a current injection the same way that we did before, instead of just asymptotically approaching a steady state membrane potential, you'll see this spiking behavior, which is happening because you have opening and closing of your ion channels depending on the membrane potential of the cell. In the Hodgkin-Huxley model, there are 3 hidden parameters that describe how ion channels are opening and closing, usually called Mn and H. They're just kind of capturing the rate of current flow across the membrane through these different ion channels, and it's a product of these that effectively gives you the conductance of the sodium channels and the conductance of the potassium channels. So by going through all of the trouble of measuring these conductances for sodium, conductances for potassium, and describing the opening and closing kinetics of the ion channels, you can capture the spiking behavior of a neuron without needing to go through any tricky steps. You're just simulating how these different currents are evolving over time based on a set of parameters that you're able to measure or fit experimentally. So there's lots of ways of thinking about how neurons are spiking, and why. One question you can ask is, why go through all the detail of simulating this rise and fall of the membrane potential? Why not just wait until you hit a threshold and spike? Why even try to simulate this at all? Part of the reason is that we won't get into this today unless you do one of the bonus notebook and the hands-on session. But once we build these dynamical models, we can do these really useful visualizations of how the models behave over time. So in addition to just plotting how the membrane potential is evolving over time, you can plot the membrane potential across the activations of different ion channels. In the notebook that you guys will have access to this afternoon, I'll provide you with the code for simulating not the Hodgkin-Huxley model, but the Morris-Lacar model, which is a simplification of Hodgkin-Huxley that only has 2 parameters. If you have just these 2 parameters, then you can look at how they evolve over time, and you can analyze a phase portrait of their dynamics. So here I'm plotting the membrane potential of the neuron on the x-axis and the activation of potassium channels on the y-axis. As you'll see in the notebooks, with the dynamical equations for the Morris-Lacar model, we can compute what are called null lines, which are these orange and blue lines here. These are basically telling us what the steady state value of either the membrane potential or the potassium channel activation is for different values of these 2 things. So if the membrane potential is very low, the steady state value of the potassium channel activation is near 0, so none of them are open. As you depolarize the membrane potential, your potassium channels are going to be pushed to open, which pushes you up this curve to eventually hit a value of around 1. So that's what this blue line is doing. The orange line is telling you how the membrane potential is going to be evolving as a function of potassium channel activation. And the intersections of these lines determine critical points of the system. They give you things like fixed points, they define limit cycles, and you can use this phase portrait to analyze the stability of your system and understand how it's going to evolve over time. So in this case, this green trajectory I'm showing you here is a trajectory of the membrane potential plotted against the potassium channel activation that corresponds to this trace up here. And it's showing you that when we give a certain amount of input to the cell, you just kind of fall along this limit cycle, and your system is circling through this portion of phase space over time. If you change the input to the cell, it's going to move these null lines around, specifically the orange null line around, and it will change where it intersects with the blue null line. And that's going to give you some information about where the spiking threshold of the cell is and how you transition from giving small current injections where you just decay back down to baseline to giving large current injections where you instead do this oscillating and spiking behavior. So that's really just a preview, but there is a notebook where you go through understanding these dynamical systems models and playing around with how the spiking behavior of these cells changes as you change different parameters of the model, be they reversal potentials or gating time constants of your different ion channels. It's a very useful tool for getting a very quick summary of how neurons are behaving and how their behavior changes as a function of model parameters. So that's the biophysical part of the class. Now I wanted to talk about describing neural activity in other ways. So what I showed you was a Hodgkin-Huxley model. This makes some simplifying assumptions, but it is also including a lot of additional detail. And in general, you can think about models in neuroscience as ranging from very complex, very constrained models that are fit to a lot of biological detail to very simplified abstract models of neural activity that throw away a lot of the detail of the biophysical system and just try to capture basic properties of neurons. So on the complex extreme, you can have these complex multi-compartment models where you simulate out really how every segment of the dendritic arbor of a neuron conducts current through it, how that current flows to the soma of the cell, and how the action potential is initiated. And there are theories and tools in computational neuroscience for understanding these things. The basic one is the cable theory model, which describes the resistance of dendritic arbors to the flow of current to the cell by capturing the resistance, the conductance of different components of the dendritic arbor and modeling how a voltage perturbation at one part of that arbor is going to be communicated to the cell body. You can build these models, you can fit them to anatomical structure data from neuroscience, you can try to capture how ion channels are distributed in the dendritic arbor and how that can add these fancy nonlinearities that gate input to the cells. There's a lot of things that you can do with these biophysical models if it's the kind of thing that you're interested in understanding. At the other extreme, you might not care at all about what your neuron is shaped like, what ion channels it's expressing, what its spiking behavior is, and you might just want to summarize it in terms of how it's filtering its input. So at the far end of having a less complex model, you can summarize what a neuron is doing just by saying, here's the stimulus that I fed to my neuron or I fed to my animal, and here's how the neuron spiked, and here's a filter that shows how I go from stimulus to spiking activity. So that's the kind of model that we're going to be fitting in the hands-on session of the afternoon. But really, this is a continuum, and different models are useful for different things. All of these models are making some level of simplification. Most models, you don't want to capture every little bit of detail that's present in the biological system, and there's certain things that you're going to throw out. But the art of modeling is to know what information is important to keep to explain the phenomena that you're trying to explain. In general, you kind of want to keep things as simple as possible, but it's worth knowing that this range of options exist and that they can be used for different scientific goals. So in the remaining time, we're going to talk about different important... Oh, hello? Was there a question? No? Okay. In the remaining time, we're going to talk about different features of this point model with ion channels and what behavior of neurons this point model captures and which of those behaviors might be interesting and worth keeping when you're trying to describe how a neuron responds to its inputs. So the first and most important thing about neurons is that they always perform some sort of filtering on their inputs. So in this, just going back to our simple conductance-based model, we applied an external current to the neuron just as a step. We inject some current, we leave it on for a second, and then we turn it off. And we saw that the membrane potential didn't instantly jump to a new steady state value, but it took some time getting there. If we hadn't done a step injection, if we'd injected some noise into the neuron instead, we would see that the membrane potential of the neuron isn't perfectly tracking that noise. It's producing a low-pass filtered version of its input. In this case, it is really just that. It's a filter that's removing the high-frequency signals from its input. But in general, neurons can act as filters of their inputs and allow certain frequency ranges of their input to pass through and attenuate others or phase shift others. So when you're modeling the activity of a neuron, the first thing that you usually think about is how it's filtering the input that it receives. So when I talk about filtering, this is just a interesting diagram I grabbed off the internet talking about different types of filters. If you have any arbitrary signal that's being fed into your neuron, you can use a Fourier transform to break it down into a set of frequency components. Frequency representations are usually helpful for thinking about time series data as kind of a summary of what a signal is doing. And what a filter is doing is it's attenuating the contribution of particular frequency components to a signal. It's removing some and it's letting others through. So there's different families of filters that you'll see in control theory or electronics. A low-pass filter is going to attenuate out any signal that has high frequency components to it. So if you make a plot of the magnitude of the signal scaled to its original intensity and the frequency, you'll see that a low-pass filter passes through low-frequency signals, but it's attenuating out high frequency signals. This is what our neuron is doing on this previous slide. It's allowing through this step response to the current injection, but it's attenuating out a lot of these little wiggles that are happening on top of that. And the degree of attenuation is going to be determined by the membrane time constant, so this capacitance times resistance term. You can construct other kinds of filters. They're less common in biological systems just because, as you saw from the model, this low-pass filtering is just kind of what naturally falls out of describing the opening and closing of, or the flow of ions across the cell membrane. In electronics, you can have high-pass filters that remove out low-frequency components of signals. You can have bandpass filters that only allow through input at a particular frequencies, and you can have notch filters that remove out signals at a particular frequency. In neuroscience, you most often see neurons that act like this, but you will also see some neurons that act like this. In some models, people really put an emphasis on bandpass filtering. When you hear people talking about like phase synchrony and phase locking and neural oscillations, what they're talking about is neurons that have a balance of ion channels such that the membrane potential of the cell is kind of resonating at a certain frequency. And if you drive the cell at that frequency, then you can evoke activity, but if you drive it at other frequencies, you can't. Oh, David has a question.
Hi, Anne. Sorry to interrupt.
No.
You're talking about filters here, and you also gave the example of the rise plateau that you see in your RC circuit as an example of a filter. But that, I thought, is determined by the membrane capacitance. That's the rise time is the time to charge the capacitor before current can flow through the circuit. And I thought that membrane capacitance per unit area of liquid bilayer is pretty much constant in neurons. What changes is the overall surface area of the neuron, depending on its size and the number of dendrites, which will increase the capacitance. So are you viewing that as part of the filtering mechanism? Or
right.
is that something different from what you're talking about here in terms of your low pass filter?
Yeah, so the filtering mechanism is the capacitance and the conductance of the ion channels. You're right, as you increase the size of your neuron, assuming that it is acting as a single unit, you can scale the capacitance just by increasing the surface area of the cell. Here, yeah, for these kinds of simplified models, you're not even really taking that into account. But if you built a more elaborate, like a cable theory model where different components of your cell had different surface area to volume ratios, you could have a greater range of capacitances, and you could use that to apply different filtering to inputs that go to like very narrow dendrites versus inputs to the soma of the cell. So you could have different filtering properties based on where an input goes to a neuron.
So can you give us an intuition about how a low-pass filter might work in terms of ion conductances and other biophysical features of a neuron's membrane?
Yeah, I guess the intuition comes from this equation that your rate of change of your membrane potential is limited by your capacitance of the membrane and by the resistance to ion flow across the membrane. So the lower your capacitance or the higher your resistance, is that right? As you change these parameters, you change the effective time constant of the membrane. And
is that every time there's a change in voltage over a particular time unit, the membrane capacitors either going to have to charge up some more, or it's going to have a partial capacitive discharge, depending on the magnitude of the shift up or down the voltage from the previous steady state. That ultimately limits the speed at which the cell can respond to a rapid change in voltage.
Yeah, the speed at which the cell responds is a function of this capacitance. The size of the current step that's injected determines the steady state value that you approach, but it doesn't, in most models, it doesn't determine the time that it takes for you to get there. That's what we see from something back here. Further back than I thought. From this here, that your rise to your steady state value is just a function of R times C, and that your value that you're rising to is a function of these other terms.
But if R for a fixed charge up the membrane capacitor before you get current flowing through the resistor.
Yeah, that's correct. So that's why the time constant is the product of these two terms. If you have a higher resistance, you're not letting ions flow through your membrane on their own. It's going to take longer for enough ions to flow through to hit the steady state value.
All right, thanks. Sorry for the interruption.
Oh, no problem. Are there any other questions? We can go back to where we were. Nope. Okay, so that is filtering by neurons, and it is a way of passing through particular signals if you have this kind of bandpass configuration of your ion channels, or just a feature of neural populations that, based on their membrane capacitance and conductances, there's going to be certain signals that they pass through very easily and other frequency components of signals that they attenuate. So in addition to filtering, another important feature of neuron models is their spike threshold nonlinearity. Neurons aren't just passive RC circuits, but they also have a threshold above which they spike. In the Hodgkin-Huxley model, that spiking is manifested by the voltage-gated ion channels. Once you pass a certain voltage value of your membrane potential, ion channels are going to start to open, and that's going to cause this runaway excitation effect where more and more positive ions flow into the cell, depolarize it, and cause this spike in the membrane potential. You can simulate this in different ways, but in general, it introduces a strong nonlinearity in how neurons respond to their inputs. So if you plot the cell activity in spikes per second as a function of the input current, you get what's called an FI curve. And this is telling you how the cell is going to respond to different kinds of inputs. And most of the time, you'll see something that is a threshold followed by either a linear function or a step function or some asymptotic approach to a maximum firing rate. This threshold nonlinearity allows the cells to, is the source of this the nonlinear transformation of signals by neural populations. In a linear system, you can only take a signal and kind of like pass it on down the line to other cells. Introducing this nonlinearity is what allows you to transform signals in interesting ways, extract out conditions such as only passing through a signal if it is above a certain amplitude. So yeah, the spiking nonlinearity is an important part of neural computation, and it's something that you can capture in models in a lot of different ways. Another feature of neuron models that is possibly important, really kind of depends on your system, is diversity of spike history effects. So if you look at neurons in different parts of the brain, you'll see different firing rate changes in response to a step input. So these are some examples. These are actually model neurons that were fit to recorded cells mostly in cortex. You'll often see cells that have this high initial firing rate and then they drop down to a steady state firing rate. These are like pyramidal cells in cortex, and you'll see them called regular spiking neurons. By changing the balance of ion channels, you can achieve other patterns of firing. You can have intrinsically bursting cells that respond especially strong to a step increase in input. You can produce chattering cells that produce a burst of spikes with a latency between them. Fast spiking cells, cells that only respond to the onset of an input and then stop spiking. And you can also create these resonator cells that respond if they receive an input at the right time relative to oscillations of their membrane potential. All of these are things that you can see neurons doing. To some extent, it's unclear when these spiking properties of neurons are important and when they're not. In some cases, the timing of spiking and whether you have adaptation or not can affect things like your ability to produce synchrony of a neural population and have sort of epilepsy-like events. These models in general or these different spiking patterns in general can all be captured by a fairly simple neuron model, the Itzikevich model, which just describes the membrane potential as a quadratic function, or the change in membrane potential is a quadratic function of its value. with this additional sort of memory parameter that keeps track of your spiking history, this U term here. So U is what facilitates this adaptation of spiking or gives you this chattering behavior. So you'll see this in some neural models when you're focused on specific cell types where you know that this kind of spiking activity is present. But it's often just kind of neglected in neural population models, and you tend to just go for every neuron is identical, it receives input, and it spikes when it crosses a threshold. But it's important to remember that these spiking properties exist, and they could be used by the brain to do computationally interesting things. Yeah?
This model is empirical, right? So
yeah.
For all the parameters are empirical, is it possible to use Hodgkin-Huxley, which describe the receptors and the membrane properties after these higher frequency or not?
Yeah, so there is an empirical component to Hodgkin-Huxley too. The time constants of these ion channels are just kind of measured. I'm not sure if you could get all of these response types with a Hodgkin-Huxley model. You probably can. This model is making some simplifications. It's not actually simulating the action potential event itself. It just has this kind of hard-coded threshold where if your membrane potential crosses a value, then it resets to a value and you increment this memory parameter. But yes, that's a good question. I'm honestly not sure how much people have tried to fit these with Hodgkin-Huxley models. Okay, so you can decide what you want to keep and what you want to throw out in your neural models. One of the most common things that you'll see in neural network simulations is a leaky integrate and fire cell. So in this case, we are tossing out the variable resistors and the spiking mechanisms of cells, and we're just saying that the change of membrane potential, we're even throwing out like the existence of a minus 60 millivolt resting membrane potential. Usually you kind of treat these as signals that arbitrarily vary between, say, 0 and 1. The only thing that this model captures is that in the absence of external input, your membrane potential decays down to, in this case, 0. And if there is input, then you increase based on the magnitude of this input current. And the rate at which you increase is set by a time constant, which is approximating this resistance and capacitance of the cell membrane. In order to get these leaky integrate and fire cells to spike, you have to hard code in a spiking threshold. And this is done very practically just in your simulation code. You say, if the membrane potential is above a threshold, then the cell is going to spike, and I'm going to reset the membrane potential back down to some baseline value. And this is actually what I used to generate this figure over here. So it behaves like the RC circuit unless you depolarize it enough to cross its threshold, in which case you kind of like add in this instantaneous spike where the cell's membrane potential is reset back down to 0. The only reason that you actually see this peak is because I added a line of the code that said set the voltage on the previous time step to be very high so that you see it as a spike. Otherwise, you would just see kind of this sawtooth rise and fall behavior. So these, yeah?
Is there a resistance term that's missing?
It's looped into this time constant tau.
But that's like tau dB dt is...
Oh, yeah, you could scale the input by the resistance.
And minus dB is also a voltage term. So
Yeah, to be fair, this I should be scaled by the resistance here. Yeah, this is like the lazy version where current isn't really meaningful units, it's just something that's being fed into the cell. Integrate and fire neurons are pretty useful for very simple simulations. They capture a lot of the properties of neurons. Like they kind of effectively give you a refractory period of cells because you reset your membrane potential to a very small value. You can do a lot of interesting simulations just with integrate and fire cells. And if you don't know much about the physiological properties of the neurons, then it's a pretty safe way to go. You can also simplify things even further, and you can ignore the spiking mechanism altogether using a firing rate model. So again, input current should be scaled, but the membrane potential evolves the way it did before. And on top of that, you have a firing rate of the cell, which is a nonlinear transformation of your membrane potential. So this is saying whatever my membrane potential is, the higher it is, the higher my firing rate is going to be. And I'm just going to predict what that rate of spiking is as a function of the membrane potential. Spiking isn't going to reset the membrane potential. It's just going to keep track of how high the potential is. These nonlinearities are typically sigmoid functions or even hyperbolic tangent, which is a little bit weird because if this membrane potential gets negative, your firing rate becomes negative. The reason that these are widely used in neuroscience is that they're very well behaved and differentiable, and they are kind of inherited from physics, where people were using models like this to describe collections of charged particles and how they interact with each other. Typically, this external input isn't just like an injected current to the cell. You'll use a firing rate model if you want to simulate what a population of interacting neurons is doing and look at how the dynamics depend on the structure of connectivity between cells. Sorry, you're frowning. Is something? Yeah? No? Okay. Yeah. Yeah, I'm almost done. So firing rate models are really a physicsy thing, and they can give you very interesting intuitions for how networks of neurons are behaving. So this is from a paper by one of my graduate lab mates, looking at if you have a population of cells and you change two parameters of the network connectivity, the strength of the connections and the degree of self-coupling, how the firing rates of cells evolve over time. And you can simulate the system with different parameter values and see settings where activity in response to an input decays down, regions where you get chaos, and regions where you get transient chaos converging to a non-trivial fixed point. And with these kinds of firing rate models, you can also often simplify things and analytically solve for where these transitions between different dynamical regimes exist. So you're throwing out a lot of information. It's not clear how biological it really is, but it gives you interesting properties, and it kind of captures the spirit of what a pool of neurons are doing. They're transforming their input in a nonlinear way based on their interconnectivity. David has a question.
Yeah, can you say something about the settings in which the previous type of model, I guess you call it a spiking model?
Integrate and fire?
Yeah. Well, you use a number of different words in talking about models. You used point models, spiking models,
yeah.
weak coding models. Can you sort of sort those out in relation to these two classes of models that you're talking about here, and when you would or would not use those different types of approaches?
Yeah.
I understand that the rate models are good for looking at population dynamics, but the other class is what you're calling point neuron models that are spiking.
Right, so point neuron just says that you're not simulating like a dendritic arbor and the filtering of inputs by that arbor. You're just treating the cell as a point. It has a membrane potential. It's not like different membrane potentials at different parts in the cell. So an integrate and fire neuron is a point model. A rate neuron is also a point model. The Hodgkin-Huxley neuron is also a point model because all of them aren't simulating the different voltages that you get across different parts of membrane. Integrate and fire model.
When would you use these spiking models that are collections of leaky input, where the points are described by this leaky integrate and fire equation as opposed to simply
a rate model? They have different behavior. I think if you care about structure of spontaneous activity, you might use, well, yeah, that's a good question.
The kind of thing that someone like E. Barter uses when she's trying to describe the influence of different neuromodulators on circuit activity. Is it more appropriate to use a leaky integrated fire neuron
instead of a
STG than just a rate model?
Yeah, rate models really are very simplified, and they're something that has kind of morphed into. Firing rate models were a concept from physics, and it's morphed into the study of recurrent neural networks, which has morphed into its own separate thing. It's not something that you would use very much if you're trying to fit biological data sets because it doesn't take into account the actual spiking of neurons. it more just gives you general population level properties. For something like you described, like Eve Marder's work, you would use either a leaky integrate and fire cell, or you'd go in even more detail and actually simulate the opening and closing of ion channels, depending on if that's something you cared about or not.
Now, you don't have any terms in these models, as far as I can tell, for synaptic weight synaptic effects.
Yeah.
So one of the things that you get, for example, out of the Drosophila connectome, is you get the precise number of synapses that a given neuron is making onto each of its targets, or that are made onto that neuron.
Yeah.
So what is the formalism that would allow you to incorporate that kind of information
Yes.
into, say, a leaky integrated fire model of such a value?
That's a good question. Yeah, so I've been kind of treating it mostly as single neuron. If you had a population of these, instead of I external being this injected current, you would replace it with a synaptic conductance. So you'd have a weight matrix between cells. If a cell spikes, then any cell it gets input from gets a transient increase in current injection that decays over time. So what you'd really have is just a second equation pretty much of the same form, which describes how the current injection at that synapse is evolving over time. And usually it's like an increase and then an exponential decay down.
But you have to take into account the number of synapses, because certain connections, if they have a high number of synapses, they may allow the postsynaptic cell to go above threshold for spiking. That would seem to be an important feature of such a model.
Yeah, there's a lot of work that's been done in populations of firing rate and integrate and fire cells, looking at how you should scale synaptic weights as a function of the number of synapses in order to achieve reasonable behavior of the cells. Yeah, I kind of skipped over the population level stuff, but that is the thing. I'm gonna skip forward in the interest of time. That's pretty much it. Yeah, I think all I had left was just a preview for the afternoon. So going back to this question of how much detail you need to describe neural activity, should you be modeling ion channels? Should you be modeling spikes or firing rates? As David alluded to, an important feature of neurons in the brain is that there's lots of them, and that these models were mostly fit based on a neuron that you're patching onto where you're controlling the current input. Most of the time, you can estimate all of these conductance parameters and these Nernst potentials. And the thing that's going to be the biggest challenge is this external input to the cells. So figuring out what input the neuron is getting from other regions of the brain, from how it's being driven by the animal's behavior. And this is what's kind of fueled a push away from these more biophysical models towards descriptive statistical models, is not really knowing what this input is makes fitting these parameters carefully a little bit redundant, because you really don't know like what inputs the cell is receiving from other neurons in the brain most of the time. So this afternoon, we're going to talk about the encoding of information by neurons. You have a stimulus feature that is translated in the brain to the spiking of a cell through many, many successive layers of nonlinear spiking neurons. And we want to understand how the neuron's activity reflects the stimulus, in spite of the fact that we don't know a lot of the layers of processing of that stimulus by the time we get to that neuron. I'll skip through this in the interest of time and just say that after lunch, we'll be talking about these neuron encoding and decoding models and how we can learn a little bit about the mapping from stimulus input to recorded spikes. And I'll wrap up there because I think I'm running into coffee. Any other questions?
Hey, Aaron, can I ask a question?
Yeah.
So I am a little bit confused about like when we model a neuron, the majority of the things we want to model is the intrinsic property, except for the current input. Mm-hmm. Is that correct?
For these kinds of models, historically, they were developed based on like slice recordings of a cell, and the input was whatever the experimenter was feeding the cell, like whatever current they were injecting with their electrode. So I guess.
Yeah, so, sorry.
Oh no, go ahead.
So I just wonder, like in the model, I saw like all the parameters except for the I external, which is controlled by the experimenter. All the other features are the major things, important things we want to model, which are the intrinsic property. Because this is kind of a follow-up question for David's question, like the synaptic inputs for a neuron is different, but this is not a feature that we can be modeled in the model we are using now, is it?
Yeah, that's kind of one of the conflicts of neuroscience is, yeah, you don't know what input the cell is receiving most of the time. If it's like imaging in a neuron in a behaving animal, you see how the cell is firing and you see what the mouse is doing, but there's this giant gap in between of how actions are translated into a specific cell spiking in a particular brain area. And I think that's part of the challenge of doing theory and computational analysis when you're looking at cells that are a little bit more removed from the periphery. So these kinds of models were very helpful for simple neural circuits and for understanding the basic properties of neurons. But when you get to the point of wanting to understand a neuron somewhere deep inside the brain, you kind of have to focus less on specific conductances and reversal potentials and instead think about just as an abstract nonlinear transformation what a neuron is doing to its input. Just what we'll talk about this afternoon. Yep.
I have a follow-up question. So you use the external one variable, but oftentimes you have like more than when you insert like current, then you get one. But in the brain, as you mentioned, like
You have many synapses, yeah.
inhibitory and excitatory, and can we model it like as, let's say we know the input, can we try and model based on like
Yeah, so
different types of like different.
these physics-based.
Repeating the question.
Oh yeah. Your question was, yeah, we're calling I external like a single thing, but a cell in the brain is receiving lots of inputs. So how do you deal with that, I guess is the question.
Yeah, do you have like several eyes, or you use like you need to model eye itself?
Yeah, so I should have had a slide on this. If you had a network of neurons, then you would have a matrix of synaptic weights between every possible pair of neurons, which would tell you how strongly each neuron activates each other. And if a neuron spikes, then you have a current injection from the presynaptic neuron to the postsynaptic neuron that's scaled by that synaptic weight. And you can play with the structure of that connectivity and the mean synaptic weight to change the way the network behaves, which is what this model is doing here. That's something I can add to tomorrow morning, I think, since I didn't touch on it today. Like what happens when you go from what a single neuron is doing up to what a population is doing.
Yeah, because you can imagine like a structure.
Mm-hmm.
You know, you have like this cell inhibitor, this cell excitatory, like let's say
Oh, yeah.
cell. What's the dynamic between these two populations?
Yeah, no, there's a lot of theory that work in that direction right now of just like what do you get if you randomly connect things together, and how do you train synaptic weights to get this population to do interesting things? That's a very rich area of development for maybe the past decade and a half or so, getting pools of interconnected neurons to do interesting things. Yeah. Should we get coffee?
Yes. So let's all thank Anne, and then we will head out. You guys have a few minutes to use the restroom, or we'll meet outside and we can start the philosophy discussion if you've gotten some coffee or water or some. All right. Thank you, Anne.
Thank you.